Okay. Here, I think we finished here. We discussed the function of the enzymes. I'm going to turn it on. But I don't think we have talked about exoenzymes, endoenzymes, regulated. No, okay, good. So that's what we're going to start talking about. So I want to remind you that on Monday we have discussed enzymes as the critical catalysts of cellular reactions, right? That are extremely specific, that are not used in the reactions, okay? And they are, what am I forgetting? Another important feature. Not used, extremely specific. Oh, they don't change the ratio. They only change the rate of the reaction, right? Now, they can be produced in various shapes and forms. Let's put it this way. We can classify enzymes based on the location they are working at. Some enzymes are categorized as endoenzymes, which means that they are produced and stay in the cell. Obviously, all enzymes are produced in the cell, but these guys also stay in the cell. You already know plenty of endoenzymes. Pretty much all the enzymes that are responsible for gene expression are working inside of the cell. If you think about it, enzymes of the aerobic respiration will be endoenzymes. On the other hand, enzymes that are released into the environment are called exoenzymes. Remember, exo means outside. Can you suggest the potential function, the possible role for exoenzymes? Role that would benefit the microbial cell. Why would microbes release enzymes in the environment around them? What would those enzymes do? Well, enzymes can't grow, but enzymes, yeah. Why enzymes are in the environment? Why they are outside of the cell? Huh? Regulation of what? What would they do specifically? What would they do in the environment? Like some enzymes break things, some enzymes synthesize stuff. And they catalyze different chemical reactions. Let's put it this way. Which chemical reactions they will facilitate in the environment? What? No. I'm talking about what will happen in the environment? What reactions will they facilitate? They are catalysts, right? Enzymes. So which reactions they will speed up in the environment? Well, growth is not essentially a chemical reaction, but you're on the right track. For growth, what do microbes have to do for growth, to grow? What's the absolutely necessary thing for us, for any living organism to grow? DNA is inside, huh? Replication's inside. In what form? No, ATP is inside, but in what form? What do we get? Where do we get energy from? What? Food. Where do enzymes get food from? The what? Where? Where do they get food from? Like location, where it is. Yeah, environment outside of them, right? So those enzymes, a lot of those enzymes are responsible for generating food, essentially. Just think about this. You have a fungus that grows on the apple. Fungal cells, they don't eat little pieces of apple. Does that make sense? They break down polysaccharides that are in the apple into monosaccharides, which then can diffuse into the cell and serve as the source of carbon or source of energy in the reactions of aerobic respiration. Does that make sense? Those exoenzymes can modify the environment so these guys can grow. Does that make sense? These guys can get the food out. Have you seen courses of like squirrels and possums and raccoons on the road? What happens to them after a while, unless they're being removed? What happens to those courses? They decay. What decays them? Microbes in the first place. Microbes don't have teeth or something to chew through. They produce enzymes that break down, you know? Does that make sense? That's exoenzyme. It goes out of the cell. They produce enzyme. They release them out of the cell. Does that make sense? It's actually pretty neat, I think. DNA polymerase, for example. Anything that's inside the cell, that's contained inside the cell, that is not released. See the difference? In many cases, actually, exoenzymes represent important virulence factor. Would you remind me, when we say virulence factor, what do we mean? Generally. Ability to do what? Virulence factor. Yes, but that is correct. We usually also we say when it's a virulence factor, it pertains to the pathogenic microbes. We shouldn't say virulence factor for rhizobium that lives on the roots of the plants, but we can say it for Staph aureus or Clostridium botulinum. Okay, virulence factor is something that increases pathogenicity of the microbe, that increases the disease, the chances to get it or the severity of the disease. So how this can work? The best example, I think, of exoenzymes are the exoenzymes produced by the microbe called Staphylococcus aureus. Just to remind you, it's the gram-positive coccus. What did I just do? Okay, coccus, right? Staph aureus produces a whole bunch of exoenzymes that can affect human tissue. One of enzymes that I want to mention to you is protease. I was trying to pick one. It has like 5 or 6 exoenzymes. Protease. So what do you think is that are protease? What do you think protease would do functionally? Break down protein. Very good. Now, Staphylococcus aureus is the common infection of the respiratory tract, but most commonly, Staph aureus occurs on the skin. Does anyone remember skin anatomy? So what 2 major layers skin has? Epidermis and dermis. Very good. Which one is the epithelial tissue? Which one is epithelial tissue? Which one? Epidermis and dermis. Which one is epithelial? Epidermis is epithelial, right? On the surface. Now, epithelial tissue, any epithelial tissue, is separated from underlying connective tissue by the basement membrane. Pretty much a layer of proteins and glycoproteins that attach epithelial layer to the connective tissue under it. The protease that Staphylococcus aureus produces breaks down the proteins in the basement membrane. What do you think will happen with epithelium when the basement membrane is destroyed? You can give me the wild guess. Exactly. This is called scalded skin syndrome. Yes. You can look at the microscopy picture, the micrograph and you can see the destroyed basement membrane and epithelial layer just slowing off. So you had an adult patient, I suppose, but it's more common in children, especially newborns. In newborns, it's not a frequent disease, so it's not like RSV or influenza, but it happens regularly in infants at a certain rate, of course, and by itself it's not horribly dangerous, but it exposes the newborn at the risk of acquiring secondary infection because skin becomes very vulnerable. It's treated with topical and oral antibiotics fairly easily and effectively, unless it's methicillin resistant, and even in this case it can be treated. But that's just one example of the enzyme leading to the certain pathological effect. That makes sense? Another way to classify enzymes is based on the regulation of their production, their making. Enzymes can be constitutive, which means they are produced at a constant level. Levels of substrate or levels of product do not change the enzyme production. Does that make sense? You have an enzyme that is always produced at the same level. All right? And enzymes can be regulated. They can respond to, for example, change in the substrate levels, or they can respond to changes in temperature or pH or whatever you can think of. Okay? You see the difference between constitutive and regulated enzymes? Now, one thing that we're going to focus on in terms of regulation is the different ways to activate or inhibit the enzymatic activity. And first I want to talk about inhibition, because if you think if you can meddle with enzymatic activity of microbes, you can certainly meddle with the virulence factors of those microbes. The two major ways of inhibiting enzymatic activity is competitive and non-competitive inhibition. They are very simple. Competitive inhibition is when the substrate and inhibitor compete for the active site. Inhibitor may be as effective in binding as the substrate, but since inhibitor concentrations are extremely high, does that make sense? It will just overcome the substrate, or inhibitor may even be better at binding the active site than the substrate. Of course, scientists can chemically design the inhibitors, and now with the current techniques, we can actually make not a database, but sort of a collection of chemicals, and then screen them for the inhibitory properties in a very rapid manner. So we don't have to spend 2 days on one chemical. It would be a bit of a problem finding the right one. Okay, so competitive inhibitor essentially, if say Michael and I would compete for the chair. Okay, whoever jumps on it first wins. Does that make sense? Non-competitive inhibition is when the inhibitor binds to another site, which is called an allosteric. Allo means other. Steri means place. So another place. Okay, it binds to another place, not active site. So as you can imagine, even small amounts or modest amounts of the non-competitive inhibitor may completely shut down the enzymatic activity no matter how much substrate you add, because substrate does not remove the inhibitor from the inhibitory site. Does that make sense? Now that would be if, say, Michael wants to sit on the chair, but I occupy that one, and I'm so repulsive that Michael just cannot see it across the table from me. Okay, that would be, I'm not doing anything, I'm not meddling with this chair, but I'm right there and it blocks everybody else from, sitting at the same table. Does that make sense? Yeah, you would be a substrate, I would be an inhibitor. Yeah, and the table would be an enzyme. Mm-hmm. Say it again. Mm-hmm. Inhibitor interacts with its own binding site on a completely different part of the enzyme. So it does not, it has nothing to do with active site at all. It doesn't even remind anybody of the active site. But it has to be very specific to that allosteric site here to bind to it perfectly. Because when it binds, it essentially changes, in a second, changes the shape of the enzyme. So it actually, if you think about this, it hijacks and changes the lock. So, yeah, it changes the lock and the substrate cannot interact with active site anymore. Teresa, was that your question? Sure. So competitive, you have a binding site. Let me erase it here. You have a binding site. But the trick is that both substrate and inhibitor can bind to it. And if the amount of inhibitor is much higher than the amount of the substrate, it will simply overcome. Or if the inhibitor is much more, let me throw a word at you, affine, has higher affinity. Affinity means how well something binds. Okay? So high affinity means that inhibitor binds extremely well and extremely tight and nothing can kick it out of the binding site. So if inhibitor has higher affinity to the binding site than the substrate, then you can use even small amounts of competitive inhibitor. One of the examples, again, remember I mentioned the abstract concepts? Now I can give you an example of the inhibitor that you may get prescribed for the disease. Have you heard about sulfa drugs? Like sulfamethoxazole, other sulfonamides? Those are competitive inhibitors of one of the enzymes, which name escapes me. I don't know, it's some dehydrogenase or something. Enzyme in the metabolic pathway for synthesis of folic acid. When we will get to the antibiotics, just striking how similar sulfa drugs are to the substrate of that enzyme. They look very, very, very much the same. And you can just overcome the folic acid synthesis with the sulfa drugs. Okay. Now, same way, by allosteric interaction, the enzyme can get activated. There's some molecule, small molecule, that makes enzyme, for instance, more efficient, okay, or actually makes it work. This is called allosteric activation, when some activator molecule binds to some other site rather than binding site. Oh, it's just not inhibition, it's activation. Other than that, it's all the same. Conceptually, it's the same principle. It's just not inhibiting, it's actually activating the enzyme. You may say, wait a minute, but if something, how activator can bind to the active site? Well, there are enzymes that require, for instance, presence of magnesium or manganese or zinc in the active site. So if you remove it, the efficiency will drop significantly. If you add it back, the efficiency will increase. Okay? And that brings us to the concept, yes? Active site. Yes. For allosteric, absolutely. Allosteric, different place. Amanda, question? Say again? I misspoke. That's active site. I meant active site. I'm sorry. Yeah. So when I say, I apologize for that. Okay. So molecule, whether inhibitor or activator, binds to the site other than active site. Allosteric, different place. So that brings us to the concept of the apo and holo enzyme. I used that analogy, but that was quite a pathetic analogy. So apo enzyme cannot work without some sort of a cofactor or coenzyme. It's like Batman and Robin, but it turns out it's quite a pathetic Batman which cannot do anything without Robin. Okay, so this is Batman, this is Robin. In order to fight a crime, in order to do the function, apo enzyme and cofactor have to come together to form a functional holo enzyme. And you can have sort of a mnemonic rule here. Holo is a whole. Okay? Like the whole thing. What can serve as the cofactor? Coenzyme, any ideas? I didn't tell you that. Any guesses? Kind of mentioned on pause. What do you think can serve as the cofactor coenzyme? No, it would inhibit. Antibiotic wouldn't serve as a cofactor. It would inhibit the enzyme. Ever heard the name acetyl coenzyme A? Coenzyme A. So different organic molecules, actually, vitamins, pretty much all the vitamins, coenzymes. Okay, a bunch of ions, coenzymes. For instance, I think all proteins that bind DNA, no matter what they do, but if they bind DNA, they gotta have zinc. DNA polymerase, protein enzyme that synthesizes the new DNA strand, gotta have magnesium. Just have to have it. It doesn't work without it. Okay? So vitamins, ions can serve, other organic molecules can serve as cofactors. Does that make sense? Now, how enzymes can be regulated? Well, first of all, we already learned about operons, right? They can be regulated at the genetic level. The level of transcription, using operons or riboswitches, or at the level of translation, again using riboswitches. But they can also be regulated, the activity of enzymes can be regulated at like the higher level, the level of functional enzyme. Imagine that you have an enzyme here, enzyme 1, that converts the initial substrate into the intermediate A. Then you have enzyme 2 that converts it into intermediate B, and enzyme 3 that finally converts it into the product. Example of such pathway would be glycolysis. Okay, you start with the glucose, you end up with a pyruvate, but you go through like 1,6-glucose biphosphate, 1,6-fructose biphosphate, then diacylglycerol, and then finally have pyruvate. Okay, a lot of hoops to jump through. Does that make sense? So you go through different steps. You finally have a product. Well, in some cases, accumulation of the product will inhibit the initial enzyme. Does that make sense? It's a negative feedback regulation. Typical negative feedback. Okay? Moving on. Metabolism as we know it. That's the material that you probably have learned before. I do hope so. So we're going to start with 3 major steps of aerobic respiration. The first step that is pretty common for all types of metabolic pathways is glycolysis. Okay, what do we start glycolysis with? Glucose, yeah. So now, one thing that I really want you to know, the carbon movement. I don't care that much about hydrogens or oxygens, but carbons, I think, are quintessential to understand the metabolism, the metabolic pathways, the energy generating pathways. Okay, so you start with a 6 carbon molecule of glucose, and first, in order to gain something, you have to invest something. Okay, so you spend 2 molecules of ATP to phosphorylate glucose. A few more reactions, you end up with a 6 molecule fructose, which is phosphorylated. Okay, now why do you do that? Any ideas? Well, not you, but why cells does it? Why phosphorylate? Why convert it to fructose and add phosphates? Nobody asked this question before. Why? Why don't we just split the glucose into the pyruvate? Here's the thing, you have to invest some energy. So fructose biphosphate can more easily split in pieces, releasing high energy electrons. Okay? It's not essentially the activation energy idea, but it's sort of similar idea. Okay, fructose diphosphate is broken into 2 3-carbon intermediates, which you see here as glyceraldehyde 3-phosphate, G3P. Okay, does that make sense? Okay, and then I'm doing the overview, and then glyceraldehyde 3-phosphate is oxidized to pyruvate. Now that oxidation, the electron that comes from glyceraldehyde 3-phosphate is captured and carried by NAD. So when NAD gains that electron, what happens to NAD? When molecule gets the electron, it is reduced. So NAD is reduced while glyceraldehyde 3-phosphate is oxidized. Does that make sense? And the final product of glycolysis is the pyruvate. Okay? That makes sense? At some point, during this step consists of many reactions. One of the intermediates there is so-called phosphoenol pyruvate, okay, which contains phosphate. During one of the steps, this phosphate is transferred to the ADP molecule that gives us pyruvate, not phosphoenol pyruvate, but pyruvate, and the ATP molecule. Now, here's the thing. We start with 1 molecule of glucose. We invest, quote unquote, 2 molecules of ATP to phosphorylate glucose. But in the end, the yield of ATP is 4 molecules at the end. So what is the net yield, net production of ATP in glycolysis? 2. We invest 2. We gain 4, the net is 2. Does that make sense? Now, is that the most important outcome of glycolysis? You can make both. If you make a point saying, so Hayden said no. Defend your point. Why? What is the most important outcome of glycolysis? So you throw pyruvate into the Krebs cycle. Good, you defended your point. Anybody else? Anybody else? Anybody who thinks that ATP is the most important? I'm gonna play devil's advocate, okay? What if there's no freaking oxygen? Pyruvate's gone, right? That's it. It's glycolysis. So in anaerobes, ATP, that's what you get. That's it. 2 molecules of ATP, period. Okay, but Hayden is right. At the scale of aerobic respiration, the amount of ATP that's produced during glycolysis is not negligible, but very small compared to what we can make. And essentially, pyruvate and electrons are the major outcome of glycolysis. Does that make sense? Okay. And NADH, because look, if you see NADH, what should you think about right away? Electron. NADH will be formed in anaerobic. We will talk about their fate, but you think about electrons. NADH means high energy electron that is carried somewhere. Does that make sense? So the outcome of glycolysis comes up to ATP, 2 molecules, NADH. So it's a reduced form, the form that carries high energy electron. Okay, actually 2 electrons, if you think about it. NADH, 2 of them, and pyruvates, 2 of them. How many carbons did we start glycolysis with? How many carbons do we have at the end of glycolysis? 6. We didn't lose any yet. Okay? Does that make sense? Awesome. Krebs. Holy crap. By the way, those pathways, glycolysis and Krebs cycle, give us perfect examples of enzymatic systems that are organized. Very rarely, enzyme works all by itself. Does that make sense what I just said? Like you have a certain enzyme that converts substrate to product, and substrate is not connected to any other intracellular processes, and product is not connected to any other intracellular processes. You see what I'm saying? Like you have a craftsman who goes into the forest, cuts the trees, makes something, and then sells it, and it's a final product. In most cases, if you look at the big industry, one company produces things that other company uses to produce its own things. So it's a chain, right? So these metabolic pathways can be of different shape. Glycolysis is linear. Krebs cycle is quite surprisingly cycle. Okay? There can be different arrangements of the pathways. Now, Krebs cycle. What is absolutely necessary condition for the Krebs cycle to happen? Oxygen. There's no oxygen, there's no Krebs cycle. We'll talk about it. So far, we presume that the microbe that we talk about is, in terms of oxygen requirements, is aerobic. Okay? The microbe can carry out oxidative respiration. Okay? So in the presence of oxygen, okay, the pyruvate, okay, becomes oxidized. Do we have that step here? No. Well, I will have to picture it myself. Pyruvate becomes oxidized. So pyruvate, how many carbons? 3. It becomes oxidized and forms, well, acetyl-CoA. How many carbons? Well, 2 that we are interested in, right? We started with 3, now have 2, unless it's a nuclear reaction. Where would this extra carbon go? Huh? And what does it form when it binds to oxygen? There you go. That's simple. Okay. Right? That makes sense? It is also, now, when carbon is oxidized, what does that mean in terms of electron flow? What's taken away from carbon? Electrons. Where do they go? Well, excellent. Some of them go to oxygen, but there are a lot of electrons that are taken. Some of them will go. There you go. So you've got another NADH formed. Does that make sense? Per 1 molecule of glucose, how many acetyl-CoAs are going to be formed? 2. Why? Why? Why 2 acetyl-CoAs per 1 molecule of glucose? So yeah, but what are they arranged into at the end of glycolysis? 2 pyruvate molecules, right? So you have 2 pyruvate molecules. Each pyruvate is converted into the acetyl-CoA. Does that make sense? Okay. Now acetyl-CoA can now enter the Krebs cycle. Okay. So I have 2 carbons here. It combines with oxaloacetic acid. Now you can say, wait, oxaloacetate, what does that mean? So I can either say oxaloacetic acid, or the anion of that acid will be oxaloacetate. Like you can see pyruvic acid and pyruvate. Does that make sense? I kind of use that interchangeably. So oxaloacetate and acetyl-CoA, C4 and C2, when they're combined, they're going to form how many carbon? C6. Okay? It's a citrate. That's why sometimes the cycle is called citric acid cycle. So C6, isomerized into the isocitrate, is oxidized to C5. What is the extra carbon goes? CO2, and you got NADH. C5 is alpha-ketoglutarate, is oxidized to C4, succinyl-CoA. The extra carbon goes to CO2, and you have more electrons produced. Okay? Then you have, here you have a step when succinyl-CoA and coenzyme A is split, that energy provides for the synthesis of 1 molecule of ATP or GTP. They both can serve as the energy source later. Okay. Then succinate is oxidized to fumarate, but at this point it's not carbons that are oxidized, it's mostly hydrogens that are oxidized. Okay. If you remember organic chemistry, no, you don't remember organic chemistry? No, pretty much. If you have carbons that are linked by a single bond, like this, okay? If you make them like this with a double bond and remove 2 hydrogens, this is oxidation, for example. So you see you have the same number of carbons, right? Single bond to double bond, it's oxidation. You have the same number of carbons, right? But you take electrons from them. Does that make sense? Okay, so now then you have several steps when succinate is oxidized consecutively to fumarate, fumarate to malate, malate to oxaloacetate. Okay, number of carbons doesn't change, but electrons are taken away from the molecule. So let's trace how many NADH. Here, 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 and there's FADH2. So during Krebs cycle itself, 3 molecules of NADH and 1 molecule of FADH2 are produced. And also, during this step, you have another NADH molecule produced during the transitional step. Does that make sense? Okay. So, transitional step gives you NADH. Krebs cycle itself, 3 NADH, 1 FADH2, 1 ATP. How many Krebs cycles are happening for 1 molecule of glucose, if I can say so? How many 1 molecule of glucose products of its degradation during glycolysis? How many Krebs cycles? 2. Exactly. Each pyruvate molecule from here initiates, quote unquote, initiates its own Krebs cycle. Does that make sense? Okay. Which means that for 1 molecule of glucose, Krebs cycle will yield Krebs cycle stage for 1 molecule of glucose. How many ATPs? 2 ATP. How many FADH2? 2 FADH2. And considering the transition step, how many NADH? 8 NADH. Does that math make sense to you? 3 in Krebs, 1 in transition step, 4 multiplied by 2 numbers of Krebs cycle, you get 8 NADH molecules. Does that make sense? Awesome. There was something else I wanted to tell you. Oh, this C3 here, what it is that enters the transitional step? Pyruvate. Now, let's take a glance at the carbons of those pyruvates. So it loses 1 carbon during transitional step. It loses another carbon when isocitrate is oxidized to the ketoglutarate. And it loses another carbon when alpha-ketoglutarate is oxidized to the succinate. So essentially, by the end of Krebs cycle, all 6 carbons of that initial molecule of glucose are converted into the CO2. Does that make sense? Now, let me remind you something. The reaction of oxidative respiration. Glucose plus 6 molecules of oxygen gives you 6 CO2 and 6 molecules of water. We found where does CO2 come from? Okay. Now we need to figure out where does water come from. We'll get to that. We're not going to get to that before we'll discuss the implications of the Krebs cycle for the metabolism of the bacterial cell. It's not only a step in oxidative respiration, it also provides an enormous number of intermediates that cell can use in different biosynthetic anabolic pathways. For example, citrate and acetyl-CoA can be used for the synthesis of lipids, like sterols or fatty acids. Alpha-ketoglutarate can be, via the step of the glutamate, used for the synthesis of amino acids and nucleotides. Succinyl-CoA can be used to synthesize complex organic molecules like heme, which is not that interesting for microbes, but porphyrin and chlorophyll. Porphyrin and chlorophyll are important pigments. Okay, chlorophyll is light-absorbing pigment in photosynthetic microbes. Oxaloacetic acid via the aspartate step, aspartic acid step, can be used again for synthesis of amino acids and nucleotides. So essentially it provides, Krebs cycle provides sufficient resources for the microbe to synthesize virtually anything it needs. Make sense? How many ATPs have we made so far? Oh, wait, how many? Wait, ATPs. Yeah, I know. How many ATPs have we made so far? Okay. How many in glycolysis? 2. How many in Krebs cycle? 2. So we got 4. Does that make sense? Why 4? Okay. I'm talking one molecule of glucose. Okay. How many NADH? 8 only in a Krebs cycle and transitional step. 10 NADH, 2 FADH2s. Which means we have a shitload of electrons. Not just electrons. High energy electrons. And remember what I told you. Energy flow in the cell is primarily transfer of electrons from molecule to molecule in the oxidation and reduction reactions, redox reactions. Does that make sense? So we have that enormous reserve of electrons. What are we going to do with it? We're going to make it work. We're going to run it through the electron transport chain. Electron transport chain consists of a number of proteins, which most of them are called cytochromes. And one of the cytochromes is cytochrome 480, 450, cytochrome 450, and that was the best license plate I've ever saw in Ohio. I saw a car with a license plate. Cytochromes are usually CYT, so somebody had a license plate like this. The nerdiest license plate I've ever saw. Okay. So those cytochromes are like steps of the ladder. Okay, imagine that you have a ball and you are at the top of the staircase. And you release this ball and it starts to jump from the ladder to ladder. By the end by the end of the staircase, it gains some momentum. Okay, it carries energy with it. Right? And in this case, what happens? Electrons have energy in the beginning, and as they go through this chain of cytochromes, they give energy away. They lose it. Does that make sense? With every transition from the cytochrome to the cytochrome to the cytochrome to the cytochrome, electrons get less and less energy. Now, if they lose energy, it has to be used for something. So these proteins that electrons travel from one to another, so essentially one protein, like this one, is being oxidized when electrons leave it, right? And this one is reduced. And then this one is oxidized and this one is reduced. You agree with me? You agree with me? So as electrons move through the electron transport chain, the energy that they give away at each step is used by those same proteins to pump hydrogen across the membrane. Okay? When hydrogen ions go across the membrane into the intermembrane space, okay, in bacteria though, well, we'll get to the bacteria, right? It goes into that intermembrane space. What's going to happen to the hydrogen ion concentration, proton concentration here in this space? What's going to happen to hydrogen ion concentration? It's going to go up. Perfect. What's going to happen to its concentration here? It's going to go down. What are we going to create across the membrane? If concentration of hydrogen is higher on one side than on another, what do we call this? Wait, not yet. We get there. We're getting there. First of all, what? When you have high concentration, low concentration, between them is the concentration gradient. Great gradient. Now this gradient is both chemical, right, because hydrogen ions are chemicals, and electrical, because you have more positive charges here than here. So those pumps, by pumping hydrogen ions across the membrane, create electrochemical gradient. Where do they take electrons from? Remind me. The proteins of electron transport chain. NADH, FADH2. So when they take electrons from NADH and FADH2, what happens to those two guys? Are they oxidized or reduced? They are oxidized. Very good. Okay, so reduced forms, NADH and FADH2, are being oxidized at the electron transport chain back into the NAD plus and FAD. Now, these two guys, NAD plus and FAD, where do they go? Huh? Correct, back to Krebs cycle and back to glycolysis. Okay, they've been reused. Does that make sense? All right, we created the gradient. So, I agree, it's awesome. It's just fancy. What are we going to do with that gradient? What gradient every gradient tends to do? It tends to equalize itself. Does that make sense? To equalize, can protons go directly through the membrane? Why? They charged. Terrific. Does that make sense? They are charged. Charged particles cannot cross the membrane. So they gotta have some sort of an opening. Right? This opening comes in the form of extremely complex molecule called ATP synthase. It's a little miniature molecular motor. It actually has a moving part, like a rotating part. Okay, so when those protons diffuse through the ATP synthase, and they simply diffuse, that's what Chastity mentioned, okay? Energy of that diffusion, which I represented here sliding down the slide, okay? Energy of that diffusion will be converted into the chemical energy of the gamma bond of ATP. So ADP and the phosphate will be combined to form ATP. Does that make sense? So this is where protons go. Okay, what happens to electrons? They go through all the electron transport chain like exhausted, not high energy anymore. Where are electrons? They have to be attached to something. You can't reduce electrons, they just electrons, but what accepts the electrons? What's the final electron acceptor? Terrific, brilliant, oxygen. Oxygen is the final electron acceptor. You have something else that is not really good for the cell. Some other particles. The protons, the acid. Where would proteins go? Protons, sorry, not proteins. Protons. What will accept protons then? Huh? What did you say? No, go ahead. I think you said no, they're going to be together, but electrons will not accept them. If they're going to be together, they're going to stick together to what? Electrons and hydrogen ions will stick together to oxygen. And when hydrogen ions will stick to oxygen, they will form water. Does that make sense? So oxygen is reduced by those electrons, and hydrogen ions conveniently bind and form water. We figured this is coming from. Cool? I think it's pretty neat. Okay. Now you have to appreciate that it's the diffusion of protons, which is also called chemiosmosis, that directly fuels the ATP synthesis. Okay. Of course, it's electrons, yes, but electrons not directly give it the energy to synthesize ATP. You gotta have protons being pumped. Does that make sense? Now, intermembrane space in eukaryotes is very simple. Mitochondria, right? You have outer membrane, you have inner membrane, you have matrix. What about prokaryotes? Do they have mitochondria? They don't. Where would you find such a convenient compartmentalization in the cell? So you gotta have it. You gotta have this whole entire system on the membrane too, right? So this system, the cytochrome system, the respiration system, will be on the plasma membrane. Okay? So that will be, in prokaryotes, it's going to be a plasma membrane. Okay? Okay? But protons are going to be pumped in gram-positives between plasma membrane and the cell wall. Same with gram-negatives in that little tiny periplasmic space. Does that make sense? I can tell you even more. So we talked about oxidative respiration. Why it's oxidative? Because it uses what? Oxygen. It uses oxygen as the final electron acceptor. It is also called aerobic respiration. Does that make sense? So if oxygen is the final electron acceptor. There are organisms, I'm not talking about bacteria, I talk about eukaryotes, that are anaerobes, ultimate anaerobes, absolute anaerobes. You have heard about some of them, I suppose. Have heard about Giardia, the infectious agent that causes diarrhea. It's the obligate anaerobe. Ever heard about Trichomonas? Trichomonas vaginalis. That's the ultimate anaerobe. Well, guess what? They don't have a certain organelle. Guess which one? They're anaerobes. They don't have one organelle. They cannot use oxidative respiration. Eukaryotes that cannot use oxidative respiration would not need what organelle? Exactly. They don't have mitochondria. Not kidding, they don't. It's pretty neat. They kind of got rid of it. It's of no use. That make sense? So that's your summary of respiration. Now, yes. We'll get there. We'll get there. We'll have. So you may have heard about anaerobic respiration as the one that produces lactic acid. We're gonna extend this concept. Okay. Now, one thing that so what you have to know. I'm gonna calm you down a little bit. You have to understand the major steps, like 6 carbon glucose to 3 carbon phosphorylated intermediate to pyruvate. What is produced during each step? ATP, NADH, CO2. Is that produced or not? Okay. What happens during transitional step? So C3 to C2. Where does carbon go? Okay. In the Krebs cycle, I don't really care that much now about the individual molecules in the Krebs cycle. Like, I will not ask you. What I will ask you, or may ask you, when acetyl-CoA and oxaloacetic acid are combined, they produce, you have to know citric acid. You have to know oxaloacetic acid. Isocitric, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate. No. I'm not going to ask you those intermediate steps. Oxaloacetic acid and citric acid, know them, know how many carbons. You got it? Products. Know the products. I can ask you how many NADH molecules, reduced forms, how many NADH are produced during Krebs cycle per 1 molecule of pyruvate. Not considering transitional step. You have to tell me 3. Does that make sense? Okay? So that's the summary. The total number of ATP molecules in this book, it's 34. If you open another Anatomy and Physiology, it will tell you 36. Some other books will tell you 40. Some other books will tell you 32, which I think is a little bit annoying. My point, it's 30 plus. Definitely not more than 40. So I may ask you how many ATP molecules can aerobic respiration produce. I will not force you to choose between 32 and 34. You will have to choose between, say, 40 and 20. Okay? So understand the ballpark. Does that make sense? I'll make it very clear on the question. Okay? Like 5, 10, 20, 40. We're going to overview respiration and fermentation, and we'll take a break then. Respiration and fermentation. Every type of metabolic pathway that yields energy, so essentially every type of metabolic pathway that produces energy is dealing with high energy electrons. So these electrons must, in the end, be accepted by a molecule. The final electron acceptor. In aerobic respiration that we just discussed on previous 3 or whatever slides, the final electron acceptor is oxygen. Does it have to be oxygen always? The answer is no. Why oxygen? Well, let's stop for a second. Why oxygen? Why it accepts electrons? Why it cannot be hydrogen or sodium? No, it's not about the charge. Good. The chemistry, yes. This bitch, right? The electrons come to the molecule or to the atom that has highest affinity to electrons, which is called essentially electronegativity. Oxygen is the second most electronegative element. The first is fluorine. Okay. Oxygen is the second. So it has such a craving for electrons that it pulls it to it. Which means it's extremely oxidative, which you can see when things rust or burn. But not only oxygen is extremely oxidative, there are other compounds like, what is it going to be? Nitrous acid, NO3 minus. I was trying to erase it with my finger. This, sulfuric acid, which are also extremely oxidative, very oxidative. Now, can this acids, essentially this anions, accept electrons? Sure, they can. So nitrate, sulfate can serve as final electron acceptor. Does that make sense? Bless you. Does that make sense? So they will be reduced. Nitrate will be reduced to nitrogen. Sulfate will be reduced to elementary sulfur or to hydrogen sulfide. Okay? But the point is, if these guys, nitrate, sulfate, carbonate, for God's sakes, are electron acceptors, do you need oxygen? No. So this will be anaerobic respiration. So a final electron acceptor is inorganic molecule, but not oxygen. It's anaerobic respiration. Now I'm going to ask you a hard question. In terms of the efficiency, ATP production, anaerobic respiration, will it be more efficient than oxidative, less efficient, or equally efficient? That's a really hard question. That's a challenge. What do you think? And can you defend your point? Will it be more efficient in terms of ATP production, less, or the same? Well, guess. I will not. Huh? Same. Less or the same. That is more correct answer. Now, remember I told you about the idea that the electron flow, high energy electrons going from the higher energy level to lower energy level. So that's your electron. Okay? As it goes from some molecule here to molecule here, okay, it will release the energy. Remember we discussed it. Okay? Now, this energy, in case of aerobic respiration, is what? The one that accepts electrons in the end. Aerobic respiration, oxygen. What's about its electron cravings? Super high, yeah? So, that craving, the ability to pull electrons and accept them, is what determines the slope. The higher electronegativity is, the more steep slope is going to be. And if you think, if the slope is steeper, the energy released will be higher. Does that make sense? So those nitrate, sulfate, carbonate ions, they are not as electronegative as oxygen. So in that case, the slope, hypothetical slope, will be not as steep, and the amount of ATP that will be synthesized will be not as high. So usually anaerobic respiration can approach aerobic inefficiency, but not come to the same level. Does that make sense? Now, can organic molecules serve as the final electron acceptor? The answer is sure. Okay. One of the simplest is when you don't have oxygen and the pyruvate becomes the final electron acceptor. Okay? It will become lactic acid in case of lactic fermentation, or can become acetaldehyde, which is then going to be reduced to ethanol. So in this case, acetaldehyde becomes the final electron acceptor, and ethanol is the final product, and that's going to be alcoholic fermentation. Well, we eat products of lactic fermentation on a regular basis. It's things like buttermilk, sour cream, cheese, and alcoholic fermentation is wine or beer, any alcohol-containing drinks. Okay, does that make sense? Now, this fermentation does not require oxygen. Okay, that's important. So there's no oxygen needed. And anaerobic respiration doesn't require oxygen. Okay, essentially, other than the lack of oxygen, the electron transport chains for both aerobic and anaerobic respiration functionally the same. They're going to be different in structure. That's there's going to be different enzymes, different cytochromes, but functionally they perform the same thing. They take energy away from electrons. They pump hydrogen, okay, in the periplasmic space, the membrane, intermembrane space, and hydrogen diffuses back through the ATP synthase right here. It's all the same. Okay. Now, where would you expect anaerobic respiration to be? Look, it's the same process but no oxygen. Where would you expect microbes that don't use oxygen? Where would you expect them to be, to live? But there is no what? Oxygen. Yeah, in oxygen-poor environments, where there's a lot of things like nitrite and sulfate. Vents at the bottom of the ocean. Okay? Now, some microbes cannot even use oxygen. What's the problem with oxygen? Oh, microbes don't really care about this. They don't know how to play with matches. But what does it do? Oxygen. It oxidizes. Yes, it produces so-called reactive oxygen species. Okay? Does that make sense? Reactive oxygen species. You probably have, I can tell you, you have a bottle, well, many of you have a bottle of reactive oxygen species solution in your cabinet. Huh? Say again? A bottle of reactive oxygen species solution in your cabinet where you store hydrogen peroxide, one of the reactive oxygen species that is formed in the cell. Others include superoxide anion, hydroxyl radical, and a bunch of other stuff. They are very reactogenic. They destroy things in the cell. You can ask, why the hell do we make them? What's the point of making them? Turns out they can destroy not only cellular structures, but also pathogens in the cell. So they're kind of useful, but not very deliberate in their action. So reactive oxygen species should be timely neutralized. And the enzymes like peroxidase that neutralizes hydrogen peroxide, or superoxide dismutase that neutralizes superoxide anion, when they are present, You see what? When they are present, microbe can deal with oxygen. If they are absent, those reactive oxygen species will kill the microbe. Does that make sense? In some cases, microbes simply do not have the gene that transfers electrons from electron transport chain to oxygen. Transfers electrons from electron transport chain to oxygen. There's no gene for it, okay? Gene for the enzyme called cytochrome oxidase. So there are multiple. So if you think about categories, first, no oxygen in the environment. Second, no functional electron transport chain. Third, inability to deal with reactive oxygen species. Okay? And, for instance, this will be obligate anaerobes, microbes that are killed by oxygen. Exactly, like treatment for gas gangrene, which is called by Clostridium perfringens, which is obligate anaerobe. When it establishes itself and it actually makes endospores, that's how people get infected. When the gangrene starts to develop, at some point it can be treated by the hyperbaric chamber. When oxygen penetrates, oxygenation of the tissues kills the microbes. But, you know, the good portion of tissues is already dead. So it's sad anyway. Let's take a break and